Welcome to part two of our prokaryotic features in our cell module. Right now we'll talk about glycocalyces and a little bit about biofilms and bacterial growth before we move on to eukaryotic features. This part can take a little bit, and so it's important to know glycocalyces as a cell structure for prokaryotes. So we're actually going to do a capsule stain in the lab, as we alluded to in our last lab. Um, and what this means is we're going to actually visualize through a negative stain by staining the background and by staining the cells. We're actually going to see this clear layer that doesn't really get stained, called a capsule. Okay, this capsule is composed of uh, like sugars in a highly organized layer. Okay, it can be sugars, it can be proteins. They call them polysaccharides, and if they're organized layers and nice sheets, it's going to form a nice capsule like we see in the picture right here. All right. If instead you have less tightly organized spaces, you will form a slime layer, and those slime layers are evident like on old driveways or in the back of sheds where there's a lot of moisture. That's a good example of slime layers that are visible. So glycocalyces, glycocalyx is the singular. Capsules are organized, just like if you were taking a Advil tablet. It's nice and organized, and if you had slimy, it's going to get it all over your fingers, and you're going to want to get it off of you. Not really organized, right? So kind of keep that analogy in your head when you're thinking about glycocalyces. We can also use them to get us started to form biofilms. You don't need glycocalyces to form biofilms, but it is important to think about. In biofilms, they are really important in things like catheters in hospitals because these biofilms are really hard to get rid of. They're a lot like endospores, but they are groups of bacteria and different types of cells that create these layers uh, of themselves, and so they all kind of share responses to chemicals, and so it's really hard to actually destroy all biofilms, and that's why they're actually a big concern in healthcare. And the big keyword would be catheters, um, and this is also um, what they do. They also prevent that those layers that are stacked up. They also prevent desiccation. Maybe the top layer gets desiccated, but all the rest will never dry out. All right, so we'll talk about biofilms one more time in nutrition and growth, but keep an eye out that the glycocalyces, the capsules and slimes, help make biofilms as well. So this is another key point of bacteria. We've kind of gone over it again, but it's important to stress when we talk about a prokaryotic cell, and that's why we're going to bring it up right here. So a prokaryotic cell divides into two. Those two become four. Those four become 8, 16, and after many hours, let's say this only took 20 minutes. So this is after 1 hour, you have this many. So after 2 hours, we're another 3 logs above. So keep that in mind. It's the old, do you want $1 million or a penny a day doubled for a month? The penny a day doubled for a month would be a lot more than just $1 million. Um, and that's the ideal growth, right? That's an exponential growth. That's going to be a nice shooting up curve. Okay. Um, but it doesn't just work like that in nature. What happens is you go through a lag phase. That lag phase is no increase in living cells, and this is starting to gather nutrients, starting to colonize. This is when nothing is really happening except chemical sensing and chemical gathering as these cells grow before they divide. Okay. Then they hit that exponential phase where every 20 minutes they'll double. That is E. coli's actual doubling time at 37 degrees. And so it'll be in this exponential growth phase. Eventually, though, uh, the exponential growth will um, cease, right? And, you know, imagine a pond overgrown. It doubles every day uh, for a month. And when is the day when it is half full? Okay, so the day before it is half full is just day 29 because it doubles every day, right? So it was half full at day 29. Um, that's the same idea here, is that eventually you'll hit a 20-minute period where you run out of nutrients. Okay, you hit the stationary phase or the uh, uh, plateau phase, and what that means is the number that you are reproducing into, 2, one of them is going to die just as fast. So this guy over here dies just as fast as 2 were made. Okay, that's this phase. We're running out of nutrients. Then you hit the decline phase. All the nutrients are gone, all the oxygen's gone, and we begin to decline. That is the decline or death phase. So you will want to commit to memory lag phase and what's happening there, right? No cell division is the key there. Log phase is active cell division. Everybody's living happily. Stationary phase, death rate equals life rate, and death phase means that the rate of new cells does not match the ones that are dying. And let's go over binary fission. Binary fission is the way that prokaryotes divide. And first you need to multiply your DNA. You need to replicate it, that's called. Then the cell actually needs to get bigger, because if you just cleaved your small self in two, you would be too small and you wouldn't quite have the resources that you need. So you have to get a little bit bigger. Then some proteins come in, FTZ proteins in this case, and start to pinch, and then they fuse the plasma membrane and cell walls together, creating 2 daughter cells. Right, that is how most prokaryotes divide and grow, and we learn about this in prokaryote chapter as well. Finally, we have some bacterial filamentous appendages, all right, and namely these include fimbriae, pili, and flagella. Okay, those are how prokaryotes can move or interact with the environment. So fimbriae are short, bristle-like, short is the key word, projecting from the cell surface by the hundreds. Okay, so they're everywhere, they're all over the cell, they are what attaches. So when you wash your hands and wipe them away, or you brush your teeth, you actually break these fimbriae and shed this bacteria so it can't attach to you. Okay, and the attaching is what is important for colonization, infectivity, and virulence. So if we can break these fimbriae, prevent them from sticking, we can get them out of here. Pilus is technically a type of fimbriae, but it can be longer, and it is typically longer, and they're not as numerous. And so you can have a pilus that forms the basis of kind of sexual transfer of genes. It's not that. It's just transferring DNA from one to the next via what we call a sex pilus or an F pilus. The DNA is actually copied as it goes into the next one, and the idea is that 9 times out of 10 or 99 out of 100, uh, the whole DNA isn't copied over, only pieces are, and that helps lend to the bacterial adaptations that we see. Flagella have two different types, okay, of flagella. These are pretty similar still, okay, but they have slight differences that you can kind of like nuance if you like. But know that they are stiff spiral filaments composed of flagellin subunits, and they have a basal body and a hook region, okay, and it spins in a motion to get them to move. That is called taxis, and if they spin clockwise, they can tumble. If they spin like this, they can, in a counterclockwise rotation, they can actually run. And that's just human terms to bacterial movement. They're only usually moving through phototaxis or chemotaxis, and then potentially magnetotaxis, which is magnets. But if there's a chemical that they like, food, they're going to go to it. If there's a chemical that they don't like, they're going to run away. And if they like light, they need light, they'll go toward it. If they don't like light, they'll go away from it. That away is negative, and toward is positive. And with that, we will end our prokaryotic lesson, part 2, and we will do the eukaryotic features in the next video.